Welcome to Biotanemy.
Next-generation sequencing. Next-generation sequencing, or NGS, is a high-throughput Sanger-based DNA sequencing technology that enables rapid and cost-effective sequencing of DNA and RNA, allowing for the simultaneous analysis of millions of fragments. Different methods involved in NGS sequencing are Illumina sequencing, ion torrent sequencing, single-molecule real-time, or SMRT sequencing. Whatever method they adopt, the NGS technique follows the same steps, which begins with sample preparation, that is DNA or RNA extraction, and library preparation. The library is amplified using polymerase chain reaction to increase the amount of material available for sequencing, followed by sequencing, data analysis, interpretation, and reporting, and finally validation. So, let's see how different sequencing methods will work. 1. Illumina sequencing. The prepared library is loaded into a flow cell coated with oligonucleotides that are complementary to the adapters that are attached to DNA. The next step is bridge amplification, where each DNA fragment is clonally amplified repeatedly to form dense clusters of identical copies, which will ensure a strong signal during sequencing. During sequencing, four fluorescently labeled ddNTPs are added to the flow cell. During incorporation, each nucleotide, it will emit a specific fluorescent signal, which is captured using a high-resolution camera. After imaging, the fluorescent labels are removed by changing the chemical conditions, like pH, or by adding certain chemicals like strong acids or alkali, preparing it for the next cycle. This process is repeated several times, ranging from 50 to 300, depending on the desired read length.
2. Ion torrent sequencing. The prepared library is loaded onto a specialized semiconductor chip containing millions of wells that are immobilized with one oligonucleotide complementary to the gene of interest. One of the four nucleotides is introduced onto the chip during the sequencing process. If that nucleotide is incorporated into the growing strand of DNA, one hydrogen ion is released, leading to the change in pH in the particular well. The semiconductor chip will convert this change in pH in electrical signal that will be recorded in real time. 3. SMRT sequencing, or single-molecule real-time sequencing. SMRT cell is a specialized device that contains thousands of zero-mode waveguides, or ZMWs. Each ZMW holds a single DNA. Into the cell, we will add four distinctly fluorescent-labeled nucleotides along with the enzyme DNA polymerase. The fluorescence will be emitted as soon as one nucleotide is incorporated into the growing end of the DNA. After each incorporation, the fluorescent label is cleaved off by breaking the phosphodiester bond between the fluorescent dye and the nucleotide.
The fourth step of NGS is data analysis. The resulting reads are either aligned to a reference genome to identify genetic variants or are assembled de novo for new sequences.
The final output of the sequence data provided as a FASTQ file is used for various applications such as genomics, transcriptomics, clinical diagnostics, etc. Findings from NGS are often validated using traditional methods like Sanger sequencing to confirm important results, particularly in clinical settings. NGS is used to sequence the 1. Whole genome to identify inherited disorders. 2. Exome, or only the protein coding regions of the genome are sequenced. 3. Transcriptome, which is an entire set of RNA transcripts at a particular stage in the cell. The whole transcriptome shotgun sequencing is used to understand any particular development and disease. 4. Metagenome sequencing, also known as microbial ecogenomics, community genomics, or environmental genomics, is the analysis of clinical samples to investigate the population of microorganisms present. The samples are not purified before DNA extraction.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.